6 minutes to complete that warm up. It is 5 questions long. Pretty simple stuff. A quick review of what we talked about yesterday related to carbs and lipids. If you weren't here yesterday, don't fear. We will talk more about it when the warm up is done. But go ahead and get started. hopefully that was enough time to complete the warm-up. If not, obviously you have until the end of the unit to complete that warm-up and all of the other assignments that you all are getting in Unit 1. This unit will end next Friday, January 22nd. So you've got 8 days to get these assignments done. That doesn't mean you shouldn't be doing them as we go through them. I don't want anybody to fall behind. I do encourage you to use your allotted time to knock these warm-ups and exit tickets and asynchronous assignments out. But of course, that leniency policy is still there for your benefit. Today's lesson is about nucleic acids and proteins, and this is a perfect time to have your notebook out and ready to go so we can take some strong notes. Hopefully you already have it out and you were using it to complete that warm-up. We're going to refer to the notes that we took yesterday in the beginning part of this lesson, and then we're going to add on to them as we introduce these next 2 biological compounds. Today's date is January 14th, 2021. I always believe that it's a good idea to include the date and maybe even the title of the lesson so that you can come back to it in the future and refer to it as you study. We also like to include objectives, at least one, and an essential question that we ask ourselves as we move through this lesson as the guiding light so that we have a benchmark to say, are we actually learning anything today? Can I answer this question? Do I know the answer? Have I gained this knowledge? It gives us a goal as we move through the lesson. Today's objectives: biologists will be able to compare and contrast the 2 primary types of nucleic acids, and biologists will be able to investigate the structure and function of proteins as it relates to the survival of living organisms. Would anyone like to go ahead and quickly read that essential question for us?
What is the basic relationship between nucleic acids and proteins in cells?
Excellent. So we're going to define the relationship between nucleic acids and proteins. This is an absolutely essential relationship. It's one that we will spend an entire unit studying. We're going to introduce this relationship today, but obviously we're going to go into much greater detail in thinking about it. I cannot underscore the importance of this relationship, so I really am going to emphasize it when we get to that slide. But to begin, this is a table that I believe is going to be really crucial to your studying as we prepare for our unit exam, as well as all of the subsequent unit exams, because in biology, all of our exams are cumulative, which means that you will have questions from Unit 1 on the Unit 8 test. You will have questions from Unit 4 on the Unit 6 test. So this table is really going to be a good summary of the first week of the class, essentially. If you can memorize this table, you're in a great place. But before we jump into it, I do want to mention that I had a really fun time looking at your virtual lockers last night. I left comments on everyone's virtual locker who did submit a virtual locker to me, and I learned a good deal about each of you. Okay, so we've got some amazing chefs in this class. We've got some burgeoning poets. We've got some photographers. We've got a lot of people who really enjoy reading, which really impressed me as well. Some people who like Bora Bora. That's one of my travel destinations on my list too. So maybe we could talk about it once we all get there one day. Some people who have some really good and interesting music tastes, and some people who everybody is unique in their own way. So I really enjoyed it. Some people who've got some really cute pets as well, and some handsome family members, some good-looking family members. So I'm appreciative of all that you all were able to share with me, and I look forward to getting to know more about you as the year goes on. Okay, that being said, if you did not submit that, it's not too late, and I will still enjoy looking at your virtual locker as soon as you get it to me. But we're looking at this table. So I told you all yesterday that we've got 4 biological compounds. We introduced the first 2 yesterday, carbohydrates and lipids. But I also told you that there are 4 things about each biological compound that I'd like for you all to memorize. I'd like for you to memorize the monomer of each biological compound, the elements that make up the compound, the function of that macromolecule, and several examples of the macromolecule as well. All right, so this is your bang for your buck table. If you weren't here yesterday or if you didn't take strong notes, This is what you want to take a screenshot of. This is what you want to save to your Google folder or have access to on your phone. And you can just flip to it and study when you've got some extra time. I think we can knock out these 8 boxes pretty quickly based on the notes that we took yesterday. So let's go ahead and see. And again, if you know that you're joining the class a little bit late, not a big deal, but please don't forget to send a message to this chat so that I can correct my attendance. Oh, I see Sienna, you've joined us. Thank you. All right. So box number 1, does anybody know the monomer of a carbohydrate? Hopefully everybody does, but who can tell me?
A monosaccharide.
Good, a monosaccharide, excellent. Now also here we have the word glucose. Glucose is the most common monosaccharide. That's why I've included it in this table, but monosaccharide is really the correct answer here. We've also underlined the 3 letters OSE. So glucose ends in OSE. Why did I underline those 3 letters? Yeah, they are an important suffix. Does anybody remember what that suffix means or what it refers to often?
Sugars.
Excellent. Yeah, thank you, JT. So when we see words that end in OSE, and I see DeJohn, you've given us that answer too. When we see words that end in OSE, automatically our mind should be thinking sugars. So I want you guys to get in the habit of making things easier for yourself. Come up with these patterns, find clues. That way you can just have these triggers. When you see on the test a word that ends in OSE, even if you've never seen the rest of the word before, automatically you should be thinking, this is sugar, that's a carbohydrate. And that gives you some information to build off of. Okay, box number 2. What are the elements that make up a carbohydrate?
Carbon, hydrogen, and oxygen.
Good, carbon, hydrogen, and oxygen. And I see, Abigail, you gave us that answer in the chat as well. So I'm appreciative of both you and Lisa. That's correct. Now, the special thing about carbohydrates is that those 3 elements exist in a 1:2:1 ratio. We talked about that yesterday, but that really distinguishes between carbs and lipids. So it's an important thing to know. Somebody else just showed up. Who was it? Ashanti. Welcome. Okay. Box 3, what's the function of carbohydrates? What do they do for living organisms?
Quick energy.
What's that?
I said quick energy.
Oh, yes, good. Quick energy. Thank you, Lisa. So that's really their primary function, especially as it relates to this class. Carbs provide quick energy, aka short-term energy for the cell. But in Unit 2, in our next unit, we will also talk about another important function that they provide, which is structure. They provide a structure for plant cells. Box 4, what do we think? What are some examples of carbohydrates? Let's get as many voices contributing to this answer as possible. You can send a message to the chat.
Glucose.
Glucose is one. Thank you, Abigail. What else?
Starch.
Starch. Thank you, Lisa. Any others? If you can think of a word that ends in OSE, it is probably a carbohydrate.
Fructose.
Fructose, good, that's one. What else?
Cellulose.
Cellulose, that's another one, good. All right. Then we've got glycogen as well. and I don't think anyone mentioned sucrose. But again, anything that ends in OSE, more than likely it is a carbohydrate. So those are some good examples that you all should be aware of. Okay, so that's the first row, the first biological compound. The second one that we discussed yesterday was lipids. Does anybody remember the monomer of a lipid? This is a little more tricky, I guess. Fatty acids are involved. Thank you, Janelle. Thank you, Abigail. And there's another part of it as well. So we've got 3 fatty acids, and there's something else.
The glycid backbone.
Good, the glycerol. So lipids are made up of glycerol and 3 fatty acid tails. So that really gives them the structure of a capital E. You've got that glycerol backbone, and then you've got 3 fatty acid tails that kind of stick out of it. So it looks like a capital E. What elements make up lipids?
Carbon, hydrogen, oxygen.
Same 3 elements, excellent. The difference is that that ratio is not necessarily there for lipids. What is the function of lipids? What do they do for living organisms?
They provide long-term energy.
Excellent. Thank you, Melissa. Lipids provide long-term energy. And then again, good. And Janelle adds that they provide insulation. That's important too. So when we think about lipids being fats, we can think about the insulation that is provided to large whales that have a lot of blubber, or bears that have to hibernate in cold environments. So fats provide that insulation to keep heat inside of a body or inside of any system. And then again, in Unit 2, we'll introduce, when we talk more about cells, we'll talk about the cellular membrane. Lipids make up the cell membrane as well. So that's a good thing to know. And our last box, what are some examples of lipids?
Waxes.
Steroids.
Good, waxes, steroids. Abigail tells us oil in the chat. That's correct as well. Excellent. And then phospholipids, we'll talk more about that. It's a big word. It sounds complicated, but it's really not. We'll talk more about it in the next unit. Phospholipids. So again, I love visual representations. You can look at them, you can study them, and you start to create this image in your mind that is kind of like etched in there that you can refer back to as you take your test. You're like, I know I wrote this in my notes somewhere, and you have that image, you know exactly what's there. So I love visual representations, whether it be a picture that you've drawn, a little doodle, a graph, or a table like this one. So I think this is a great thing to have copied down in your notes. It kind of summarizes what we're talking about. And by the end of today, you'll be able to go in and add these last 2 rows to complete the table. So I'm leaving it up for, let's say, 15 more seconds for folks to write or take a picture or a screenshot, whatever you plan on doing. Okay. So carb or lipid. I'm going to review some terms here, and I want you guys to just tell me quickly, carb or lipid. Let's just get through this. So the first one, waxes, carb or lipid.
Lipid.
Excellent. Quick energy, carb or lipid.
Carb.
Carb, excellent. Cell structure, which one's that?
Carb.
Carb, good. Glucose,
Carb.
carb, good, JT. Cell membrane,
Lipid.
that's the lipid, good. What is that a picture of?
Lipid.
Lipid.
Lipid, good. We've got a lot of people contributing here. What about long-term energy?
Lipid.
Lipid.
Good. Lipid provides that long-term energy. Glycogen,
Carb.
that's a carb. That's an example of a carb. Carbon, hydrogen, and oxygen.
Both.
Both.
Yeah, you can make a case that this is both. Now, how do we distinguish between the two carbs and lipids?
The one with the ratio.
Good, thank you, Sally. So carbs have a ratio, and does anybody remember what that what that ratio is?
1:1.
1 to 2 to 1. Good. Glycerol and fatty acids.
Carb. No. What's it
Lipid.
called? Lipids.
Yes, this is a lipid. Thank you, Lisa, and thank you, JT. And last one, we've got a little hexagon shape. What's that?
Carb.
Carbs.
Yeah, good. Specifically, that's glucose. That's a monosaccharide. Good job. All right, you guys, if you can really have this information filed in your head for literally from now until May, that provides you, let's say, 2 to 3 questions on your EOC exam. Okay, we're going to add on to it today by introducing nucleic acids and proteins, but this is a great start. So good job. Before we jump into the content and introduce nucleic acids, I want to ask you all this question. So we see this image. We've clearly got people with different clothes on, different skin tones, different hair textures. What is it, do you all think, that makes people so unique?
Genetics.
Excellent. Thank you, John. And what exactly do you mean by genetics? What are genetics?
Like DNA, I think.
Good. So we should be thinking already about this term, DNA. Now, you might not, I'm sure you've heard of it before, but you might not know exactly what it is or what it stands for or how it's made, but we should already be thinking about this term DNA. This is the substance that gives us all of our unique qualities. It's why no two human beings who have ever lived are exactly alike. Because even if you've got an identical twin, I have a fraternal twin. She's a female, so of course we're not identical, but my twin and I are obviously very genetically similar. If you have an identical twin, you and your twin are extremely genetically similar. But even then, there are small differences in your genetic makeup that will give you slightly different outcomes. Somebody might have a birthmark in one place, and someone might have a slightly different skin texture. Someone might prefer math while the other prefer social studies. Whatever it may be, there are small differences even between two individuals who are exactly alike. The reason why no two individuals are copies of each other is because of DNA. And we're going to introduce it today and talk a lot more about it really for the rest of our semester. So DNA is a nucleic acid, and in fact, the NA in DNA stands for nucleic acid. DNA is one example of a nucleic acid. There are other examples, but the function of all nucleic acids is to carry genetic information and to control the activities of the cell. In Unit 2, we will spend that entire unit talking about cells, and we'll come to find out that the cell is a really complex, complicated place. We can think of it as like a large mall. And in this large mall, like maybe you've been to North Lake Mall or you've been to Concord Mall. In this large mall, obviously there are a lot of stores, there are a lot of employees, there might be a food court, There are patrons who are there to actually shop. You've got all these different activities going on. The nucleic acids in the cell are there to control those activities. They are comparable to the owner and the operator of the mall all together in one person or in one role. They say this store has to be open from 7 a.m. until 7 p.m. This store can only be open from 9 a.m. until 9 p.m. We have to receive all deliveries between the hours of 5 p.m. and 5 a.m. The food court has to close at 7 p.m. and can only serve this type of food. The nucleic acids would be in charge of all of that. Only 500 people can be in the mall at a time, X, Y, and Z. So they control what happens inside of the cell, and they carry the cell's genetic information. The genes, they carry the instructions, the instruction manual of the cell. The monomer that makes up nucleic acids is called a nucleotide. So again, I want you all to pick up on patterns and make things easier on yourself. Start seeing things so that you can create some associations between terms. No problem, Lance. Thank you for letting me know. When we see these letters NuCle, we should already be thinking about nucleic acids. We should be thinking about nucleotides. And we're going to introduce a few other terms as well that sound very similar that should be obviously related. Nucleic acids are made up of nucleotides. The nucleic acid is the polymer. The nucleotide is the monomer. And nucleotides have three parts. They have a nitrogen base, they have a phosphate group, and they have a 5-carbon sugar, otherwise known as a pentose sugar. Of course, pent means 5, like a pentagon has 5 sides. So pentose is a 5-carbon sugar. So I think the monomer really looks like a house. The 5-carbon sugar looks like the house with a satellite dish, that's the phosphate group, and a flag, that's the nitrogen base. And we still see those same 3 atoms, carbon, hydrogen, and oxygen, but we introduce 2 others, nitrogen and phosphorus. Oops. Leaving it up for a few more seconds. The rectangle. This says nitrogenous base. I can send a message to the chat. Nitrogenous. Okay. If you need me to go back at any time, just let me know. So here are the 2 primary examples of nucleic acids, DNA and RNA. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. So the difference here is that DNA is deoxy. So RNA has an oxygen atom that DNA does not have. DNA is deoxified. It's deoxified. And ribo refers to the sugar, ribose. So DNA has the ribose sugar without oxygen, and RNA just has the ribose sugar with oxygen. That's just a little bit of extra information. DNA is double-stranded. So it has the structure called a double helix. It looks like a spiral, two ribbons that are kind of wrapped around one another. And DNA has the nitrogenous bases, the nitrogen bases, A, which stands for adenine, T, which stands for thymine, C, which stands for cytosine, and G, which stands for guanine. Now, you all don't need to know exactly what they stand for. You just need to know the first letter. Yes, Janelle, yeah, you're thinking totally on the right track. We'll talk about that in a little bit. RNA, on the other hand, is single-stranded, so it looks like just one singular ribbon. And it has, instead of T, instead of thymine, RNA has U, which stands for uracil. So even within RNA, there are different types of RNA as well, and we'll talk more about some of them. There's tRNA, mRNA, rRNA. There's also what's called mitochondrial DNA. So there are some other types of nucleic acids, but the two general classes are DNA and RNA. Okay. So here they are again. DNA on the right and RNA on the left. We can see that DNA has thymine and RNA has uracil. You can see a very, very small difference between them. Thymine has what's called a methyl group. Those of you who are interested in going to med school or becoming nurses or just doing medical research, you'll probably end up taking a class called organic chemistry, in which you'll study the actual shapes of these compounds. So I'm not going to complicate it right now, but you can see if we were to take these two pictures, thymine and uracil, and compare them side by side and say, what's the difference between them? Well, the difference is right here. You can see that thymine has what says H3C. Really, it would be CH3. And that's what's called a methyl group, methyl, a methyl group. So it's a very small difference. There's only 4 additional atoms that create the difference between uracil and thymine. But it ends up making a pretty big difference. Also, we can obviously see that RNA is single-stranded and DNA is double-stranded. But in any case, both of them are absolutely essential for all living things to function. You will not find a living thing, regardless of if it's a human being, an elephant, a rat, an insect, a sea urchin, a bacterium. It doesn't really matter. All living things have DNA, and all living things will use RNA in order to make proteins, which is what Janelle is talking about. And we'll talk about in a couple slides. Before we do that, let's see if we can compare and contrast these two nucleic acids. So as you see, we've got a word bank at the bottom of your screen. I'm going to move from left to right. And I want you all to tell me where these terms go. Do they go on the left side of the Venn diagram with DNA and the right side with RNA, or do they go in the middle, meaning they apply to both? So let's see. Deoxyribonucleic acid, which one is that?
DNA.
Excellent. Thank you, Abigail. Single-stranded, which one is that?
RNA.
RNA. Thank you, Stelly. Double-stranded, which one is that? Thank you, Melissa. Has uracil. Which one has uracil?
RNA.
RNA, good. Now uracil really, you guys don't need to remember that. Just know that it's the U. RNA has U. Which one is found in all living organisms?
Both.
Both of them are, yeah. Which one has thymine?
DNA.
DNA. Thank you, Lisa. And again, you don't need to know, or you don't need to memorize the word thymine, but we can refer to it as just T. DNA has a T. Nucleic acids, which one, where should that go?
Both.
Both of them. Ribonucleic acid.
RNA.
RNA. And then lastly, which one of them is made of nucleotides?
Both.
Both of them are. Good. Trick question there. All right, so I'm going to leave this up so you can do some comparing and contrasting. You can write it in your notes. Again, I love visualization. Venn diagrams, really simple, but they work. Okay. So this is the point that Janelle was making. DNA is directly related to proteins. Now, she brings up a term called ribosomes, which is really advanced. I love that she's thinking in that way. We'll talk more about ribosomes in Unit 2. I'm not going to mention them here, but DNA, this is the most important sentence that we'll talk about today. DNA provides the instructions to make proteins. DNA provides the instructions to make proteins. So I always like the analogy of a cookbook or a recipe book. We've all got our own favorite foods. I saw some of you are quite the chefs already. I saw some pesto chicken that I was ready to jump through the screen to grab in the virtual locker. But let's say we've got a recipe. We get this recipe. I think was it Melissa who made the pesto chicken? I can't remember, but someone made some really good looking pesto chicken. Let's say that we're trying to make another plate of that pesto chicken and we don't know how. So we need to start off with the recipe book. That recipe book is like the DNA. It tells us what we need to use. It tells us what temperature we should start off with. It tells us what ingredients we should combine and how we should combine those ingredients and when we should combine those ingredients. So DNA is going to tell you all of that. But in a recipe book, of course, there's not just one recipe. So instead of just having a recipe for pesto chicken, we're going to also have a recipe for pupusas, and we're going to have a recipe for mashed potatoes, and we'll have a recipe for any number of other foods, whatever your favorite, some type of pizza. I know we had a lot of pizza fans in here. So DNA is going to be a big, really thick recipe book with 20,000 or 30,000 different recipes in it. But on any given day, you might only need to make 2 or 3. So on this day, we're going to just make pesto chicken. So we just flip to the page with pesto chicken. Now we have the exact instructions of what to do. That's what DNA tells us. The ingredients that we need to use to make pesto chicken are similar to amino acids. Amino acids are the monomer that make up proteins. They are the monomer for proteins. So in my recipe book, I've got the ingredients. That's the DNA. It's going to tell me I need chicken, I need pesto sauce, I need some olive oil, I need some salt, I need some pepper. Those ingredients are my amino acids. And when I combine them all into my actual dish, then the pesto chicken, the final preparation of the pesto chicken, is my protein. So the DNA is the recipe, amino acids are the ingredients, and the meal, the dish, is the protein. DNA provides the instructions to make proteins, just like a recipe book provides the instructions to make a meal. Now keep in mind, if DNA has 30,000 different recipes in it, obviously you're not making all 30,000 in a day. Again, you might just make 2 or 3. In fact, maybe your job is only to make 2 or 3. You make those same 2 or 3 every day. That's what you wake up to do. That's what you go to sleep thinking about, making those same 2 or 3 dishes. And that's what most cells do. Your muscle cells wake up and go to sleep thinking about how they can produce movement, how they can contract for your body. They're not worried about making, melatonin, and they're not worried about making keratin and all these other things, these other proteins that it simply doesn't need. All it needs is to be able to do this. So it wakes up and goes to sleep. How can I make the proteins that will allow movement? Okay, so we'll talk a lot more about that. This is a concept that we will dedicate an entire unit to in a couple of months. But I love that Janelle is already thinking about it. This is the most important sentence that we'll cover today. DNA provides the instructions to make proteins. What are proteins? Proteins are basically everything that is you. The hormones that trigger your body, that it's time to go through puberty, those are proteins. The hormones that trigger your body, that it's time to go to sleep, those are proteins. The chemicals that give your skin color. Some of us have color year round, some of us have color in specific seasons, some of us never have color. Those are proteins. The chemicals that you taste when you bite into a juicy steak or piece of pizza, those are proteins, or salad. So proteins serve most of the functions for living organisms. In fact, most of the cell itself is made of some type of protein. They are the soldiers of the cell. They do anything that the cell needs them to do. Again, their monomer is called an amino acid. We've got 20 different amino acids. And when we connect these amino acids, they're connected by peptide bonds. So when we connect them and we chain them together, they kind of look like a pearl necklace, or maybe if you've ever seen one of those candy necklaces that you probably used to get when you all were in elementary school, and you would just bite off one piece of candy at a time, or those candy bracelets. That's kind of what peptide or polypeptides look like. They get bonded together and then they fold up. So if you've ever had a chain and you just kind of threw that chain in a bag to transport it somewhere and it got all tangled up and then it took forever to untangle, that's kind of what proteins look like. So we can see a protein folding here. Now, the way that proteins fold is not a random process. It happens in a very strategic way. But if you're interested in that, take AP Biology. It has to do with the chemical properties of these amino acids. The atoms, again, we've got carbon, hydrogen, and oxygen. So carbon, hydrogen, and oxygen appear in all 4 of our biological compounds. With proteins, we also add nitrogen. And again, as I stated yesterday, a central theme of our course is that structure determines function. So if a protein folds one way, it's going to have one function. I could take that exact same protein, meaning I have the exact same amino acids, and I could fold them in a different way and totally change the function of the protein. It's actually so specific that we can think of it like two hands on the same body. If I've got my left hand, it looks almost exactly like my right hand, but they're mirror images of each other. And so because of that, they're different. If I've got a protein and then I've got another protein that is basically a mirror image, but they fold in a slightly different way, or they move in different directions, then they're different. The structure and the way that proteins fold determines what they're able to do. Okay, so here it is again. I'm not going to stay on this slide for very long because we'll talk, like I said, we're dedicating an entire unit to this later on. But DNA holds the information to build proteins. Okay. Now, like I said, there are billions of proteins in our body. All of the cells in your body are constantly making proteins, and they make many, many different types of proteins. But the 5 that you have to know for biology are here: enzymes, which we'll talk more about, hormones, hemoglobin, insulin, and antibodies. Your asynchronous assignment today, one of them, will ask you to investigate and research briefly these 5 types of proteins. But I will leave this on the screen for, let's call it 90 seconds. Okay. All right. We only have 20 minutes left, and I do want you guys to get to this exit ticket and these 2 asynchronous assignments. So we'll come back to the Kahoot tomorrow. The 2 asynchronous assignments, of course, can be found in Canvas in our module, Unit 1, Day 3. The protein research assignment will take a little bit longer than the other one. So if you want to start with the exit ticket and then move to macromolecule matching and then do protein research, that might give you some momentum. So you might consider doing it that way. I will be here on mute if you need anything, or you can just ask me in the chat if you have any questions. Okay, folks, it is now 1:20, so I'm going to stop sharing. I'm going to stop recording. I look forward to talking with you all one more time tomorrow. If you need anything between now and then, please don't hesitate to reach out. And I hope you have a great rest of your Thursday.